Good morning. My name is Isabella Grumbach. I'm here with Andres Hidalgo from Yale University. We just concluded a fascinating session on inflammation in the cardiovascular system. Andres, I'm a vascular biologist. I study smooth muscle cells. I almost never think about neutrophils as a driver of cardiovascular disease. And you taught us otherwise this morning. In your talk, you characterized neutrophils into 3 different clones based on their behavior in the vasculature. What prompted you to do this, and what do we need to know about these clones?
Right, that's a good question. So what prompted me to study, because it's a long story, but the interest on neutrophils is an even longer story that comes from just imaging the cells inside vessels when I studied my postdoc with Paul Frenet. And then realizing many years later that what we were seeing there was not all random, which is what you think when you look at a biological system and you measure your things. So there's some cells that are high, some are low. This is just technical or it's computational noise or something, or biological noise as part of. Then I guess it was years later when we started thinking, we got more resolution to study the cells, any cell type, neutrophils in particular. But you say, well, maybe there's better ways to measure these. And when we came to realize that if we use different metrics, then patterns emerged. That's where the behaviors came, not because we were thinking about behaviors, but it's more about the technically solving the question of what was going on in the vessel. Could we report this in a better way? Proteomics or flow cytometry fail, transcriptomics fail miserably. And so as we were progressing and with the love on imaging, things sort of converged naturally into finding, oh, this is grouping the cells in a way that seems interesting that we haven't seen before. And then the associations with there's this specific pattern that we see when the animal is having a higher inflammatory injury. And then the opposite, some are reduced. Oh, there's a specific gene that we can associate with these. So as we knew more and more, the interest grew because then we realized not only it's there, but now we can characterize better. And now even we're at the stage in which we can eliminate those cells altogether genetically. And as you make the question, well, that observation that was a bit random at the time, can we actually, in a mouse for now, can we improve the resistance of this mouse to a stroke or to myocardial infarction genetically? Because now, being this type of entity, then you can now think, even in humans, so well, if this is a separate entity with different biology, different genes, different targets, maybe now I can finally do what we couldn't do in humans, which is altogether get rid of the bad neutrophils.
You identified a clone 3 among your neutrophils, which stick to the endothelium.
Yeah.
How do these neutrophils look different from other neutrophils?
They're interesting neutrophils. They're bigger, they're flatter. They like to, they don't move, and they really get stuck in the vessel. So they look morphologically, in terms of movement, very different from the other ones. And now tracing back, we know that they also look different in many other ways in the terms of gene expression, even where they come from. So that's it really seems to be a separate entity. So that in an experiment that we've done where we genetically label a type of neutrophil, so those are red, and then we look, in the vessels, and we see that most of these B3 type neutrophils are red, but not the other ones. So that's cool. It tells us that it's a separate breed of cells.
And you characterize those as being drivers in cardiovascular disease. What are the genes which are differently expressed in these neutrophils?
Yeah, well, that's hopefully something for the next paper, because we're actively working on this. But a collection of these genes are related to monocytes and macrophages. So they seem to be more hybrids of monocyte macrophage. They're neutrophils any way you look at them, with markers and the nucleus and the size, everything tells that it's a neutrophil. But it's genetically more like a monocyte. which tells you not that it's a monocyte, it tells you that it's a separate breed, and there's a collection of genes that you can think of like specific chemokine receptors, the complement-associated genes, antigen presentation genes that are expressed. And some of these are, you can find as proteins as well. So we know quite a bit about these cells now.
The other really, really fascinating part of your talk was to learn that these different populations of neutrocytes derive from different bone marrow locations. So the neutrophils that derive from my femur are different than the ones that come from the tibia.
Yeah.
That was incredibly fascinating. I've never heard this before.
Yeah.
How is this a way to develop potentially new therapies?
Yeah, who knows? But I guess all things start. Imagine with the checkpoint blockades, I guess the first experiment was, oh, look, this molecule, if we knock it out, then the T cells go crazy. And they I don't know, oh, look, the tumor is smaller in these mice. So thinking always, I guess that's the inspiration of a success story. But it's a little bit the same thing. If you realize that there's anatomy in inflammation, right? So there's good bones and there's bad bones. This is just hypothetical. There's two very clear paths that you can target. First is what is the bone-to-bone difference, that you can understand why this bone is supplying bad cells. Is it about supply, or is it about the quality of the cells? And there's no such thing as bad cells. All the cells are good. They're just bad for a specific context, right? We're not, I guess, evolutionarily designed to live the way we live in many ways now. So that's one thing we can learn about. Oh, why is the humerus producing a bad type of cells? What's special about those neutrophils? Oh, maybe other cell types that are also bad, or even stem cells producing to start expand with atherosclerosis and not with. You can start thinking of things. The evidence is, suggesting that these local expansions do occur. So not all the bones respond the same thing to the same challenges. And not that it's directly related, but it tells you that not all the bones, when we think about bone marrow, is not a uniform bone marrow, and that's all the same. That is compartmentalized, and maybe for a reason. And the other thing, so we're thinking about interesting experiments in the mouse. Imagine where we expose them to cancer or to atherosclerosis or some sort of inflammation. And then we say, well, what happens if now you locally irradiate? So you can ablate temporally. And people are finding that, for example, the skull bone marrow, it's quite different from other areas. That's an example. And that's, good and the bad is that you can almost safely irradiate the head, because most of the cells are quiescent, and the only preparative would be the bone marrow. So you can ablate the bone marrow and then ask the question that's been done. What happens? You're still alive because, the body is producing blood. But now you don't have this particular contribution of cells that are very different. So you can ask these very specific questions, and in the mouse even more so, where you can start playing with different. So very simple, and considering what therapies people use, just imagine that the specific areas of the bone or the anatomy that produce in specific conditions, again, no good or bad. For certain things they're bad, for certain things they're good, but you have a specific condition. And you know that the supply, and in the mouse and hopefully in the humans as well, the supply of cells that promote a type of tumor really is from the sternum. And that sounds like a weird thing, but that could very well be. Could you target specific bones by ablation? I mean, we do even worse things now in the clinic, where you just have this chemotherapeutic agent. It's going to mess up your entire bone marrow. Deal with it.
So this is a completely new concept of treating atherosclerosis, potentially.
For example, yeah.
To which extent do the findings from your mouse models apply to humans? Is there experimental proof, similar processes?
There's some proof that we gather in that might be, that we always think about these things. Of course, you don't think that the mice is an entirely independent species from, being mammals. I guess the basic strategies, they're the same. They have cancers, they don't have probably atherosclerosis in the wild, but they have also muscular complications. And so we always think in those terms. And specific ways in which, for the talk that I presented, we look if there's some correlates, if there are, for example, clones of neutrophils in the human. There's not a very easy way to do it if you don't have access to genetic tools like we have in the mouse. But there's a simple experiment that I presented at the end where you can take blood samples from the mouse or from the human and see if they self-organize in terms of behavior, if there's all random or there's independent of blood vessels, if they organize in 3 predetermined sort of behaviors. And if that would be the same as in vivo. So that we did in the mouse, and we see that even on plastic, they sort of group in this type of behavior. So that was very exciting to see, okay, this really is independent of the environment. This seems to be coming from the inside the cell. But now we can do it because that's easy to do. We can do that in the human. We just take blood and then do this. And that's what we've done, and so far so good. I mean, it's not published, not reviewed, so I cannot go too crazy with this, but so far it looks that they know how to group themselves. So the concept of populations of predetermined behaviors seems to be there. That's very easy to read, and that clearly is what we see. Can we tie it with, so what we're doing now, if we take COVID patients, if we take septic patients, even cancer patients, and we use that blood and see if they organize in the same way, are these some behaviors that expand, some that disappear, new ones that appear. So thinking about diagnostic as well. Can we use this pattern, which has quite a bit of resolution, to demonstrate that humans have the same strategy as mice, and if we can use it for therapeutic ideas, yeah.
That is really fascinating. So there are many new possibilities based on your findings.
Yeah.
Well, thank you very much for sharing your really exciting findings with us this morning. I think we're all very much looking forward to hearing more about the possibilities of targeting neutrophils in cardiovascular disease and beyond.
Yeah, we're very excited. Thanks so much for having me. Thank you.
Of course.